Welcome to today's MAST webinar. I think we should get started now. So our speaker today is Emma O'Halloran from the University of the West of Scotland, who will talk about developing techniques to study Neoparamoeba species isolated from sea urchins. A couple of housekeeping notes to begin with. After Emma's presentation, we'll have a live Q&A session. So if you'd like to ask a question, please just use the Q&A box that you can see at the bottom of your screen. You can submit a question at any point and we'll work through them after the presentation's finished. So this session is scheduled to end around 1:30 p.m. UK time, but we may go over due to questions. The entire webinar will be recorded. So if you miss anything or you can't stay for the entire session or you want to share it with a colleague who couldn't join today, you can find the recording on the MAST YouTube page along with all of our previous webinars as well. So a quick introduction to our speaker. Emma is a second-year super doctoral training partnership postgraduate researcher at UWS. Their research focuses on Neoparamoeba virulence. I probably should have practiced that out loud before. Its unique microbiome and impact on amoebic gill disease and gill health in salmon. She achieved a BSc in biology from UWS in 2021 and has experience working at Glasgow Scientific Services. The work Emma is presenting was recently funded by the Saltire Emerging Researchers Scheme, which was a joint collaboration between the Scottish Funding Council and MASTS. So without further ado, I'd like to hand over to today's speaker, who is Emma.
My name is Emma O'Halloran and I'm a second-year PhD student studying at the University of the West of Scotland. I'm going to speak today about my exchange to Tenerife, where I focused on developing techniques to isolate Neoparamoeba species from sea urchins. So yeah, my PhD is looking into the microbiome of Neoparamoeba prurens and investigating its role in disease prevalence and severity. So Neoparamoeba prurens are a type of amoeba, and amoeba are eukaryotic organisms that ingest bacteria through phagocytosis. So bacteria can have the ability to evade phagocytosis and have the potential to act as an endosymbiont within amoeba. In the cases where bacteria are pathogenic, amoeba can then act as a vector for the bacteria species, and so it's important to consider this relationship when investigating the pathogenicity of amoeba. So organisms of the Paramoeba genus are characterised by their intracellular endosymbiont called the Perkinsella-like organism. This is a eukaryotic organism that's related genetically to a common fish pathogen. So because of this, it's thought that this relationship could also infect the virulence capabilities of Neoparamoeba species. The species I'm interested in is Neoparamoeba prurens. This is a species known to cause amoebic gill disease, which is detrimental to the Scottish aquaculture industry because Atlantic salmon are really susceptible to the disease. On the slide here, this is a culture that I'm working on, which was isolated from the gills of Atlantic salmon farmed in the west coast of Scotland. So the disease is characterised by excess mucus production, which leads to white mucoid patches and fusion of the gill tissue. So this leads to respiratory distress in the fish, and if left untreated, can cause mortality in the fish. So it's known that dysbiosis in the gill microbiome causes gill disease pathologies, and so it's thought that the bacteria in which the amoeba, inside the amoeba, what it harbours, can play a role in disease onset and severity, and so I aim to research this further. So the prevalence of bacteria in amoebic gill disease also causes problems with the culture of amoeba. So during the isolation of amoeba, antibiotics are commonly used to have xenic cultures, which is where there's no other organisms present in the culture. But when antibiotics are added to Neoparamoeba, the bacteria are persistent, and attempts to reduce the bacterial loads can also cause deterioration of the amoeba cells. The strain of Neoparamoeba that I'm currently working on was found to have an intimate relationship with Vibrio species. This was found during fluorescence in situ hybridization microscopy, which is in the image here, where Vibrio was found to be predominantly intracellular. So you can see in yellow here, this was a Vibrio probe. This relationship is really important because Vibrio are notorious marine pathogens and also infect humans with around 500,000 cases noted annually. There's a lot of potential for Vibrio to be an endosymbiont to amoeba because Vibrio larvae is an endosymbiont to another marine protozoan parasite, and also Vibrio tasmaniensis is an oyster pathogen, and it's been able to evade phagocytosis in amoeba species. In terms of studying this relationship further, sea urchins are a good candidate because these organisms are also susceptible to amoeba disease and Vibrio disease as well. So Neoparamoeba species have been associated with mass mortalities in sea urchins, and Vibrio are commonly isolated during bald sea urchin disease. So for this research, I was awarded a grant from MASTS and Saltire to visit the University of La Laguna in the Canary Islands. Here I worked with the Institute of Tropical Diseases, who specialise in investigating clinical and environmental amoeba species. This group also previously investigated the events causing mass mortalities in the Canary Islands. Due to problems with attaining amoeba, the objectives of this exchange were to develop a consistent approach for Neoparamoeba isolation. We also wanted to isolate Vibrio because of the highlighted relationship within the Scottish isolate, and the end goal is for any successful isolates to be characterised and compared against the isolates from Scottish waters. In previous successful isolations, they used non-nutrient agar dissolved in sterile seawater, but the amoeba commonly attach to the plate, as you can see in the image, and also float in the media. So because of this, we added a 5 ml overlay of sterile seawater onto the agar plates. We also used malt yeast agar dissolved in sterile seawater because this is commonly used in amoebic gill disease isolations. And before beginning the experiment, we also prepared some TCBS medium, which is a selective medium for Vibrios. We were provided with 21 samples by Dr. Jose Carlos, and these were of 4 different species of sea urchins. They were gathered and delivered to the lab on the same day, and as there were no current disease outbreaks, the urchins were presumed healthy, except for one urchin that displayed signs of spike loss, which is a common indicator of disease. So this is Ferrichinus granularis. This is Paracentrotus lividus and Orbatia lexula and Diadema africanum. So the coalomic fluid, we chose to screen the coalomic fluid for amoeba because this has a similar function to the blood in higher animals. So in the cases of disease outbreaks, pathogens are usually found here. To attain the fluid, we used a 1 ml syringe and inserted it through the peristomal membrane, which is surrounding the teeth. We holded this at an angle so that it would avoid contact with the lantern area. So from here, we added 300 microliters of the fluid on each agar plate, and then added the 5 ml of sterile seawater overlay. Additionally, added the algae from the sea urchins into Page's amoeba saline solution, which is just a common media used for the growth of amoeba species. This was just to check if there was any amoeba present in the environment. And the plates were checked daily because the plates are prone to bacterial and fungal overgrowth, which can jeopardise any amoeba present on the plates. Upon inspection of the amoeba, I found that there was some things that looked like possible amoeba. So inside the coalomic fluid, there's various immune cells. In this image here, there's phagocytes, vibratile cells, and also amoebocytes. In this image, I thought this was Neoparamoeba just because of the way it's floating in the agar and the shape of it. But when I researched it, phagocytes also look like this in phylopodial form, which is an important shape change in the clotting process. Here In this image, I think we have pigment cells, which are another immune cell that clump together and become rounded during stimuli from the immune system. So even though I didn't see any promising signs of amoeba, I still continued with the methodology to try and isolate anything of interest. I firstly carried out replicate plates, which is where you cut out a area of interest in the agar and transfer it onto a new plate, and then amoeba should move around the new plate. During this step, dead bacteria can be added and placed in the new plate so that the amoeba move towards the bacteria. I also tried a pellet plate, which was where I centrifuged the overlay of the plate with the sterile seawater and then streaked it onto a fresh plate, with the same idea that amoeba movement would be easier to notice if the plates had less debris and bacteria on them. Because there was a lot of bacteria in the overlay, I also tried to put the chlomic fluid just onto dry plates to see if this would prevent the bacteria build-up and ease microscopy. And then lastly, I tried to cut out pieces of agar and put it into clear flasks with a flat bottom in liquid medium MYB. So this is usually how the Neoparamoeba species are cultured, but it's quite costly, which is why I left trialing this till the end. So for the isolation of Vibrio species, I took the chlomic fluid and put it on TCBS agar and left it for 24 hours at 37 degrees. Due to one urchin showing signs of spike loss, the outside, the exterior of urchins were also swabbed and streaked onto plates, and we done this for 2 urchins per species. So for Sphaerochinus granularis, 2 out of the 4 samples had a lot of vibrio growth, and 2 separate colonies of both yellow and green were noticed. This species contained the sample with spike loss, but interestingly, the sample with spike loss was negative for vibrio internally and externally, and the other external swab was positive. For Paracentrotus lividus, the 5 out of the 7 samples had presence of vibrio growth. However, it was limited to a small amount of colonies. 4 out of the 5 positive samples had 2 separate colony morphologies, and the other just had a yellow colony. From the exterior swabs, only 1 again showed a yellow colony and the other was negative. For Arbacea lexula, 5 out of the 5 samples were positive for vibrio, and 3 out of the 5 had extensive growth for yellow colonies. From the external swab, again, 1 sample showed the presence of a yellow colony. For Diadema africanum, 2 out of the 5 samples had presence of vibrio growth. 1 was positive, showing yellow colonies only, and the other had, again, 2 colonies. From the external swabs, there were no vibrio on the plates. So overall, the internal vibrio presence varied among the species, and a range of colonies were witnessed, but notably, Arbacea lexula, all the samples were positive, and were positive for 2 different colony types, and Diadema africanum had the least vibrio presence internally with no exterior swabs. It was also noticed that the exteriors of the urchins, if they were positive, only had 1 or 2 colonies, so the vibrio was much less prevalent on the exterior than inside the chlomic fluid. Conclusions regarding both the amoeba and vibrio require molecular characterisation. This is necessary, firstly, to identify if the amoeba species were in the chlomic fluid and the isolation trials were just unsuccessful and the methods weren't fit for purpose, or if there were no amoeba in the chlomic fluid because there was no disease outbreaks. So molecular characterisation for vibrio species as well will give species-specific insights, and this information can highlight any trends in commensal bacteria within the urchins. So for this, we'll use specific free-living amoeba primers and vibrio-specific primers to give us these results. Technical recommendations for future experiments would be to use MYB in the clear flask to start, because although they're the most expensive route, it would save a lot of time during microscopy, and also, if the collomic fluid's added to a liquid medium, it would amplify the biomass, which would come in handy for increasing the DNA yield, which would for molecular work. I would firstly, again, prioritise the molecular work, because if any samples are present for amoeba, more attention can focus on isolating these samples, which would decrease the loss of potential amoebae due to bacterial overgrowth. I would also, I tried to grow Vibrio cultures in just liquid broth, and they weren't successful. So next time I would use marine broth or Vibrio, because I know that definitely works to grow them in liquid. And again, back to the isolation trials, I would utilise the use of antibiotics, because even though it would, or the microbial load, it would still increase the chances of getting an amoeba isolate that isn't lost to bacterial overgrowth. So in terms of impact, the work presented was a screening of healthy sea urchin samples, but it's still useful for disease outbreaks, because it gives insight into natural distribution of both amoeba and Vibrio within sea urchins, which can help highlight dysbiosis during future outbreaks. Also, the work contributed to method development, which is vital, because infections with amoeba and Vibrio are set to rise due to climate change. So having dependable methods that you can follow in the case of disease outbreaks would be beneficial. It also contributed to my own PhD training and helped give me insight into the wider problems associated with amoeba pathogenicity. So I'd like to thank my funders for the opportunity to develop my skills and contribute to research. I'd like to thank my supervisor, Professor Fiona Henriquez, for her contribution to the exchange and her support throughout. I'd like to thank Dr. Jacob Lorenzo Morales and his team at the university for hosting me, and also Dr. Jose Carlos Hernandez for providing the urchins. And lastly, Ronnie Maria for their expertise and support during the exchange.
Thank you so much, Emma. That was really interesting, and it sounds like you got a lot of work done in a short amount of time. I'm not sure if we can see you again.
Oh yeah.
Turned off the video there for people, just for the connection. So just a reminder to everybody, if you'd like to submit a question, you can use the Q&A box at the bottom of your screen, and we'll try and get around to it today. So I wondering if I can ask the question for myself. So you spoke about Vibrio species. What does it mean if they're found? What does that mean for diseases? Does it make it more likely?
So yeah, well, it's possible that the Vibrio are co-infecting on the gills or in other organisms, and if this is the case, Vibrio could be screened in the environment to alert if an amoebic outbreak could happen, or also Vibrio could be used as an indirect drug target for amoeba diseases. So if the amoeba depend on Vibrio for survival, antibiotics could use to target Vibrio instead of the actual amoeba. So yeah.
Thank you. And I know that you work on salmon, isn't it, usually? So how was it different for you to work on sea urchins?
I actually found it really enjoyable. I don't actually I've never actually isolated amoeba from gills. I've just been working with the culture in the lab, so actually getting a chance to try and isolate them from an animal was it was really interesting. I enjoyed it a lot.
I mean, I don't think we've seen a photograph of someone in the lab yet from one of these exchanges, so that was really lovely to see.
That's cool.
I would just like to thank you again, Emma, for presenting today, and for everybody who's watching. Before you leave, I'd like to remind you that you can find more information and sign up for future talks on the MAST website, and we'll also circulate it with the emails and the MAST Twitter account as well. So thank you very much again, Emma.
Oh, thank you. Thank you.
Bye, everyone.
Bye.